So again, my name is Daniel Nelson. A little bit about myself. I have my Bachelor of Science and Master of Science in Biological Sciences. Also have my Associate Arts in Mathematics. And I completed my degrees from the University of Houston Clear Lake. A little bit of fun fact about the University of Houston Clear Lake. It was originally founded in the 1970s to support the NASA space program. Very close proximity to the Johnson Space Center, Houston. And I'll kind of talk about a little bit more about that as we kind of move along.
And
So,
sorry,
what is
sorry to interrupt you, Daniel, but just a quick info for everyone. Yeah, if y'all are logged in with your ACC ID, you don't need to put your name in chat. If you want to play it safe, you can, or if you're not sure if you are logged in with your ACC ID, but otherwise it shouldn't be necessary. Apologies for the interruption there, Daniel. You continue, please.
Sure. So what is CRISPR? CRISPR is known as clustered regularly interspaced short palindromic repeats. This was a normal sequence found within archaea and bacteria and was assumed to be an acquired immunity for bacteria. An ability to recognize viruses such as bacteriophage, when they infect a bacteria. They found about to be about 40% of the bacterial genome and 90% of archaea. As far as the history goes, in 1987 in Japan, this was when it was first termed I believe I interrupted here, but interrupted clustered repeats, which would later be known as interspersed. This was studied in E. coli originally. In this case, the point of the CRISPR, and we'll talk about this in a second, has to do with more so the sequence. And what we're going to end up using in 2025 as a technology is more so going to be this Cas complex. And the discovery of that, or it's more so its importance, was more so in the 2000s. So in 2005, once again, this was hypothesized as an adaptive immunity for bacteria. And in 2007, there was evidence as this was sure enough an adaptive immunity, and bacteria were indeed having some sort of memory function when it comes to repeat infections. In 2008, there was a strong argument that indeed these Cas complexes were evident, and specifically the Streptococcus pyogenes genus and species would be the big player for these Cas proteins. This is when you typically want to study new Cas proteins, you'll do so by studying Streptococcus pyogenes. This is the going to be the genus and species that express these at a really high level. So this is kind of the standard when it comes to Cas proteins. And as we know, in 2003, the Human Genome Project would be completed. So again, it kind of all aligns with that history. So we're looking at a prokaryotic cell that's currently being infected by a bacteriophage. In this case, we see that the foreign DNA sequence is inserted into the cytosol here, to which it will actually be incorporated in these CRISPR clustered repeats segments that are going to be separated by these things known as spacers. Later on, we will give this a more specific term that we're going to use to actually target our specific gene when we want to use this as a biotechnology. Okay, so in this case, this is known as the acquisition phase. Again, this little sequence will quite literally incorporate itself in the sequence here for the bacteria, and then there will be a set of Cas proteins. Now, in order for these Cas proteins to assemble themselves, it's very specifically is going to rely on this little hairpin loop, part of this CRISPR RNA structure. And as you can see, once the assembly is complete, upon subsequent infection, this Cas protein will use the help of this RNA to seek out the complement sequence that it's looking for, and the Cas protein will act as an endonuclease. In fact, it's known as an RNA-guided endonuclease, and it's going to seek this specific sequence that it first was insulted with, okay, or first challenged with. Okay, once it does so, it simply just disrupts the genomic sequence, and as you can imagine, this will prevent this bacteriophage from being successful. Okay, so stage 2, or just into this next little topic here. And by the way, I source or cite everything that I talk about today, so if you want to look up any of these papers, you can. And this one here is just showing you again another cartoon, just shows that upstream of the Cas9 protein sequence is this tracer RNA. And there's other Cas proteins as well to keep in mind. Sorry about that, guys. Didn't mean to do that. Let me switch back there. Okay, so let me continue on. So again, downstream of Cas9 are going to be other Cas proteins as well. Again, there's Cas1, Cas2, Cas7 through 11, 13. There's several Cas complexes to be discovered. In fact, today, if anybody's looking to do a project and characterize Cas proteins, that still very much is available. Okay, but we're going to focus again primarily on Cas9 today. That's going to be the main protein that we're going to use as an endonuclease. And once again, whoops, come back here. Once again, once the virus infects this cell, the CRISPR sequence will be, or excuse me, the sequence will be associated or incorporated in the CRISPR sequence. This again known as the CRISPR repeats. And again, it's the use of this tracer RNA that's going to both help assemble the Cas9 complex, but also use this CRISPR RNA as a guide towards the viral sequence that it needs to make a cleave or cut. Okay. So again, this is known as CRISPR RNA. This will have a recognition function. The other RNA that we were looking at in that previous image is known as tracer RNA. And the tracer RNA is going to be complementary to the CRISPR RNA. And this sort of forms a complex or hybridizes together. Now, the point of the tracer RNA, again, is to help stabilize and activate the Cas9 complex, which is what we're going to be looking at as we move along. Okay, excuse me. So here we're looking at CRISPR RNA complex with the tracer RNA. From this moment on, we're just simply going to refer to this as the guide RNA. The guide RNA is just the combination of these two. So again, we're just going to refer to this whole complex as a guide. Okay. So once again, once this is expressed, this will complex with the CRISPR RNA, that is the tracer RNA and CRISPR RNA will join to form our guide. Okay, once again, this will stabilize and as we'll see in a few slides here, will enable the assembly of this Cas9 complex, which will then again seek out our target. And we'll talk about how we seek out a target here in the next couple slides. Okay, again, let me know if there are any questions as we move along too as well. Okay, so if we zoom in here, this is what we're looking for. Specifically, there's going to be a complex with our nuclease that's going to target this protospacer adjacent motif. What a protospacer adjacent motif are those little spacers that we saw in the previous slide. Now, term protospacer adjacent motif, this is a sequence of an NGG where the N can be any nucleotide in the 5' to 3' direction. So use your imagination, compared to, typical restriction endonuclease sites like Hindi1 or Hindi3 that relied on a palindromic sequence. This is a very, very powerful genetic tool. Okay, we have the ability to basically isolate two subsequent guanines and just have any nucleotide that sits in front of that. So this is a very, very powerful tool, very easy to use, and also, as I mentioned in the introduction, very inexpensive. Okay. Now, the goal here is that the protospacer adjacent motif is within 20 nucleotides of a protospacer, and the protospacer of which is 20 nucleotides in length as well, that it's going to be capable of making, not only being recognized, but inducing a double-stranded break, okay, which is going to be the point of the Cas9 system. Okay. Now we see here our guide RNA. Again, this is both the CRISPR RNA and also the tracer RNA combined. This would be the CRISPR RNA that we see here in red. But again, we're just going to simply refer to this as the guide RNA as we move along. Is going to hybridize with its target strand. Okay, now one thing to comment now is that the guide RNA will have the same sequence as the protospacer. It will just be the RNA copy or RNA version of that sequence. Okay, and so again, in this case, we're displacing the non-target strand and forming a hybrid between our guide RNA and our DNA. Okay, once we do so, it will simply make a double-stranded break, and then that's what we're going to be using as our technique here. Okay, let me just check the time as I move along here, if y'all don't mind. Okay, awesome. Okay, so here we're looking again at the guide RNA. Guide RNA is both CRISPR RNA and tracer RNA combined. Again, the point of the CRISPR RNA is to be the complement to the target sequence, and the tracer RNA is simply to stabilize the Cas assembly. Once again, the nuclease themselves will target or it will be associated with the protein that will target this protospacer adjacent motif. Again, this can be any nucleotide followed by two subsequent guanines in the 5' 3' direction adjacent to your favorite gene. Okay, so this is a very, very highly specific genetic editing technique. Here again, we can use site-specific mutagenesis or cleavage. Once we induce this cut, as you can imagine, it's going to require some type of repair. In this case, we can aim for non-homologous end joining, and as we'll look at in a second, we can hopefully aim for homology-directed repair. Okay, and again, there's some more information about this technology if any of the biotechs that are attending today are seriously considering ordering some of these reagents or tools. Again, this can be very easily done at the community college level. Okay, so again, looking here, the goal is to aim for either non-homologous end joining or homology-directed repair. In this case, the idea of HDR, homology-directed repair, is ideal. This is what we hope for, because we can aim for site-specific mutagenesis. That is, we can hopefully insert some type of donor DNA that's going to be incorporated during the time of HDR. And those who are not familiar, HDR wants to use the copy of the sister chromatid that doesn't have the genetic insult as a template to correct the mutant allele. Okay, so in this case, HDR is going to be ideal in any situation. The trouble with HDR is that it has to be done so very close or near or during the time of cell division in order for the sister chromatid or the 2N2X chromatid to be available. Okay. Otherwise, non-homologous end joining will suffice, as we'll see in a couple of research papers today. Non-homologous end joining, it's sometimes taught as it's not as good. Non-homologous end joining is very much going to be perfectly fine for somebody that's considering doing genetics from a medical standpoint. I mean, if you could just correct whatever the insult is by just putting a band-aid on it or patching it, usually it's good enough to correct whatever disease is presenting from that early stop codon, etc. So usually non-homologous end joining is perfectly okay, but as mentioned here, it can result in frame shifts or insertions and deletions. Okay, so what we're going to be looking at, this is a pretty cool paper. This is actually a pretty remarkable paper. This comes from nuclear imaging. Let me, in fact, go to my little cheat sheet here. So what this is a high-speed atomic force microscopy. Some of y'all have maybe actually seen this technology before. If you've ever seen the images of little myosin walking on actin, or the rotary action of the F1 ATPase during catalysis of ATP synthesis. So some of y'all maybe actually seen this technology before, but we're going to be looking at that for our Cas9. Okay, so a couple things just to kind of show you here before we look at the videos. As far as the nuclease or the endonuclease complex is going to be this H and H and this rough C, and it's going to use this protospacer identifier or protospacer interacting domain to recognize the protospacer adjacent motif. Now you notice there's a series of REC 1, 2, and 3. These are going to be responsible for associating with or stabilizing the guide strand. But more specifically, it has to do with their flexible position, and that's what the researchers were discovering, is that these REC proteins are very much going to be in a dynamic flux and changing their position to enable this complex to form. Okay, it's almost like if you've ever been on a crowded elevator before, everybody kind of has to kind of scoot and wedge and jam. Is very much the best way I can kind of describe the assembly of these domain. Okay, it's almost like they're scooting out of the way for H and H and Rev C to kind of tuck in and with respect to the target strand. Okay, now once this assembly is complete, what we're going to see in the image is that the Rev C and the H and H are going to fluctuate. They're going to alternate between a high position and a low seated position, and specifically when it settles into the low seated position is when H and H is being translocated towards the target. Okay, and that's when we'll see it displacing this development of this little R loop, which is indicative that your hybrid is currently forming. Okay, so that was a good thing as far as researchers are concerned. Once again, and you'll very easily see this. Once it shifts to this position here, it will induce the double-stranded break, and this is going to be indicative of the active conformation. So I believe they're going to call this the D conformation. All right, so this is what we're going to be seeing. Again, if we look at this image here um, of crystallography, they were just showing you that typically this would form a closed position, but in the presence of magnesium chloride, as we know, DNA synthesis is a magnesium-dependent process, that it would assume the active position or the D conformation. Okay, so that's what we're going to be looking at. If you'd like to, I'm going to go to the Google Drive for a second, and again, we're going to be looking at the fluctuation of this H&H domain specifically. And I'm just going to show you a really cool video. If you've never seen this before, it's pretty neat. So I'm going to take a second here. And by the way, this was published in Nature magazine, November of 2017. So this was a pretty profound project. Okay, so I'm just going to go to the drive here just for a second. Oops, wrong one. If I can figure out how to get through this, hang on. Should be this one here. I'm just gonna look at this one here. So this is the best video to look at. I titled it "Double-Stranded Break with Cas9 and Single Guide Strand RNA plus in the presence of magnesium chloride." So we see that it's currently fluctuating, and basically it's kind of settling in on that target. And the pink, gotta kind of slow down here a second. So the pink line here represents the fluctuation of the H&H domain. And then all of a sudden you're gonna notice it's gonna make a cleave. Okay, hang on. And then there you go, the cleave is made. Okay, so you can actually see in real time the double-stranded break occurring. And again, you can actually see that hype that in that last image there, you can see that fluctuation of that H&H domain. Okay, so very, very cool technology. Okay. All right, so back to this. Again, if you want to read more about that paper, it's quite fascinating paper. Again, provided you the link to that in the Google Drive. Okay, so this is what we were seeing on the previous image. And again, it's just showing you the difference between the two states of the inactive state in comparison to the activated or low position D state. Okay. So again, let me know if there are any questions. So I'm going to take the time here before we shift on to the applications.
Okay. Yes, Daniel, I was kind of wondering how initially the Cas DNA kind of inserts itself into the double-stranded DNA. Like, how is it able to do that?
Good question. So the nuclease function will actually unwind it like a helicase. It's in the function of the H&H and the so yeah, good question. So the H&H, in fact, let me go back for a second. So the H&H domain specifically is going to be making the double-stranded break on the target. The RUV-C is going to be making the double-stranded break on the non-target, and they have an inherent helicase function. So they're capable of unwinding the strands themselves. Very good question. Now, one interesting topic is there is a lot of studies on the evolutionary side of this when it comes to those that study the evolutionary side of this. A lot of interesting stuff come from that. So people are still studying Cas1, Cas2, the other Cas proteins as well. So, but that's my best answer for now, Devin.
Oh, thank you.
Mm-hmm. Okay, so moving on. So as far as some applications, as we know, over the past few years, it's if you've noticed, the idea of genetic editing is becoming more and more in the zeitgeist or popular culture, if you will. Genomic editing, the big concern was always somatic versus germline editing. Whether we're editing gametes versus editing an adult somatic cell. Over the years, we've seen, for the most part, somatic gene editing. Here in the United States, any edited embryos have to be terminated within 14 days. So, anything as far as germline is going to be definitely approved through the FDA or the clinical trials. Interesting enough, there's other applications of Cas9 as well, such as transcriptional regulators. For those of you that are considering epigenetic work or chromatin or histone remodeling, I cannot stress this, I recommend this enough. The catalytic domain can be inactive. That is, we can replace it with a forcal domain that's no longer going to recognize our Cas complex. And that could work as well. In this case, what it does is it tries to displace polymerase and will result in methylation of a target gene region. So these dead Cas9s are very, very useful transcriptional regulators. There's also Cas9 variants. This is where it has no catalytic activity. In this case, it's not going to induce any type of double-stranded break, but this could be used for things like fluorescent tagging, which again, there's already been some really, really brilliant work done with this. Use your imagination. Okay, whoops, wrong way. So again, this just kind of shows you a rundown of the different type of technologies. The one that we would probably be using, if we want to do like a protocol with the zebrafish and inserting a DNA by using some type of expression plasmid, we'd probably be using like a regular Cas9 function to induce a double-stranded break. In this case, again, the goal would either be non-homologous end joining or HDR. But interesting enough is that that Cas complexes can be also used as a nickase. In this case, in fact, a lot of the contemporary efforts are using Cas9 as a double nickase rather than inducing a double-stranded break. And there's a lot of really neat things that's emerging from that technology, specifically in the field of medicine. Then lastly, another neat use, those of you are maybe considering teaching a microbiology course at Austin Community College, you could do a very easy GFP experiment, which would give, students a visual indicator and something to kind of be excited about in the classroom, when you kick the lights off and turn on the fluorescent lighting. So, again, you can use, dead Cas9s and even include some type of a transcriptional regulator or some type of positive marker such as the GFP. Okay, and many different colors available. Okay, so this is again just showing the GFP technology. In this case, this little tag is applied to your Cas complex, and again, upon a positive indication, you'll see that it'll kind of light up bright. Okay. But again, this is not the only editor on the block, right? Some of y'all with experience in genetic editing probably familiar with our class 2 restriction endonucleases, right? We have, of course, our zinc finger nucleases and our talons, which are transcriptional activated like effector nucleases. In this case, they can also induce a double-stranded break, but oftentimes they're limited to a less specific forcal domain, which is why they're more so acting as a restriction endonuclease. Okay, so they do have their limitations in comparison to Cas9, but they still are very, very, very useful in the field of biology today. When you consider things like zinc finger transcription factors, etc. So they're not without purpose. So just something to consider. There are other means of genetic editing that are still available other than Cas9. The pitfalls of using these is they're very, very expensive and they're not always easy to use and they're not always efficacious. In the case of talons, the point of them is basically to purposely, and to induce, a frame shift, very, very prone to frame shifts, etc. So there's several pitfalls to using this in comparison to, site-specific mutagenesis in CRISPR-Cas9. Okay, some issues. Again, the pitfalls. One concern was mosaicism. So, as we know, when we do if you did not know, when we do somatic gene editing, a lot of times it requires that we give serial injections. That is, you're not going to get 100% of the human organism or species to have edited cells. So the human's going to have some cells that are edited and some cells that are not edited. And the concern was, will the body target mosaicism? Well, for my capstone that I did at the University of Houston, my responsibility was to read paper after paper after paper after paper after paper. I think I read somewhere around 70 to 80 papers in 2018, trying to get as much information and understanding of CRISPR-Cas9. And it's ethical concerns. And from what I was able to understand at the time, that mosaicism, and we'll look at several papers today, mosaicism is not of concern. That is, the body doesn't target the edited cell as being foreign. So when you think in terms of clusters of differentiation or cell surface proteins, it doesn't seem to have any type of effect. Okay, when we edit these nuclear genes. But also to consider mitochondrial studies as well. Cas9 can be used for any type of DNA, not just nuclear genes. So mosaicism doesn't seem to be a problem. Next is off-target sites, off-target indels or insertions, deletions. Single nucleotide polymorphisms is always going to be a concern. This is still sort of one of those topics that it's still kind of being debated. What the efficacy and safety of CRISPR-Cas9. I can tell you that there are FDA-approved CRISPR-Cas9 technologies that are going through clinical trials today. So from what I've read, off-target indels or off-target insults seem to be minimal. Okay, and that's a good thing. Next is downstream effects. As we know, gene transcriptome. When we look at the whole transcriptome or omics from a whole entire from a 1,000-foot view, a lot of times genes can have a downstream effect. Consider the induction process during development in our mother's womb. As we know, gene expression has an effect on other gene expression downstream. So again, we should always be kind of concerned of downstream effects or unintended effects. Next is monogenic versus polygenic genes. When we're trying to prove efficacy, we have to consider whether something is controlled monogenically or polygenic, and how effective this treatment's going to be. Also, to the delivery of Cas9, at the community college level, it'd be easiest for us to kind of just use an expression plasmid. In fact, one of the things that I actually have available is the protocol that I typically like to use in plasmid design. Very, very easy process to do. Simply just doing PCR, and with quality control, we can just use gel electrophoresis as we move through the steps. So we can use an expression plasmid. But as far as medicine is concerned, the big use is the adenovirus, or using viruses, lentiviruses, etc., to deliver the Cas9. Okay. The next is the immune response. Again, the big concern was that, would this lead to an immune response? You have to understand that Cas9 is relatively a large protein, but again, from the papers that I've read, the immune response or adversary effects have been minimal. Okay. Ethical concerns is that unintended consequences. Again, we could say tread lightly, or we could say that most fears are irrational. It's kind of one of two camps with kind of how you want to proceed with editing the genome. My recommendation is just do your reading, do your research, and do your homework before you commit. To doing some type of protocol like this. When you think about say like keeping the astronauts safe. I think editing the astronauts should be the last choice. If we can protect them with material sciences, that's probably the best choice before we have to start, doing somatic gene editing of these astronauts, right? So unintended consequences we should always be concerned about. Next is consent and parental autonomy. This was always a big concern. I can tell you that recently, one of the first clinical trials was done with an infant, and it was for a child that was lacking a liver protein that would have ultimately become fatal for that child. And this baby is living perfectly happy life, perfectly happy baby. But that choice wasn't made. That choice was made on behalf of the infant, and so we've already ripped the Band-Aid off with that. So we can definitely learn from that experience. Next is germline versus somatic gene editing. As we know, if we edit the germline, this is going to be fixed in the population, with respect to our gametes. But the benefit of germline editing is obvious. In that case, some type of fatal disease such as Duchenne's muscular dystrophy, that person is going to have a permanent edit for the rest of her life and live a happy, normal, happy life. Whereas for somatic gene editing, I'm going to have that patient come back to my clinic again and again and again for the rest of their life to continuously give them gene therapy. So it's a risk versus benefits type conversation. And then also two misleading results, the quality of the test, considering homozygous studies versus heterozygous parents, back to that risk versus reward type mentality. And again, when it comes to quality of the test, the big concern is honesty. Have a lot of these labs been being honest when it comes to, efficacy, but also, risk, okay, unintended concerns. Just check the clock really quickly here. Okay, excellent. Okay, so moving along, again, we've looked at the natural CRISPR pathway. We would be working in the realm of the engineered CRISPR-Cas9 system. This is just showing an expression plasmid, which has the promoters necessary to express our genes, both the Cas protein and our guide RNA. This just shows you a comparison of a control plasmid versus a knockout plasmid. And this on the right here, we just see things like our guide strand. We have termination signal, of course, the CBH beta-actin promoter that's going to be used to drive the expression of the Cas9. Also, we have some nuclear localization sequence, so it's going to have those basic residues required to enter into the nucleus, where it will be able to make its cut. And in this case, they included a GFP, and then a second 2A peptide, which is going to support the expression, and a U6 promoter as well. So just kind of give you the idea of how primer design works. Again, I can reassure you it is easy. It seems intimidating. It is easy. Okay. So moving on, this was just the paper that I used primarily to understand mosaicism. In this case, they were editing embryos, and the concern was, of course, mosaicism. And what they found was that, sure enough, mosaicism tends to be tolerated. Okay, that the body doesn't recognize these cells as being foreign under any condition. So some current human studies at the time, again, I did this in 2018. You have to realize that CRISPR-Cas9 studies in the US was very limited in 2018. There was like 2 labs doing any type of research back then. Opportunity was very, very limited. Now in 2025, opportunities are starting to explode for CRISPR-Cas9. So that's kind of why I'm here today. Okay, so in this case, we're looking at MYBP, familial hypertrophic cardiomyopathy. Beta thalassemia is a big one. This has heavily been studied. And of course, mitochondrial disease is a really nice argument for that. One big study is the deletion of the CCR5 gene in immune cells to confer resistance to HIV, but this has been scrutinized. This is again, a lot of this research is not going to be without scrutiny. So you have to kind of use your own kind of judgment when reading these papers. And also to the country it's coming from, impact factor. Okay, I can confirm that I try to take the highest impact factor when I put this together. So, but next is retinus pigmentosum, editing stem cells, sickle cell anemia, editing liver cells, hyperlipidemia. This is one we're going to look at today. One I'm really highly concerned about is editing of induced pluripotent stem cells. This is a technique being used to cure type 1 diabetes mellitus. Okay. But this is an outdated list, as there are several human clinical trials in 2025. There are some controversial candidates, including germline editing of monogenic diseases such as Huntington's disease. What about pre-implantation genetic devices or diagnosis? So in this case, this is another thing kind of being debated. Also, heterozygous parents versus recessive carriers. Very rare for 2 homozygous recessive parents to mate. I mean, it can happen. But typically more rare. Polygenic diseases, also to cancers, heart disease, diabetes. So efficacy will always be of conversation. But next is again polygenic continuing. Just mentions common variants, rare variants often have larger effects. Heterozygosity can also offer protection for many diseases. So again, there's a lot of things you have to weigh as a researcher or as a physician. Okay, before committing to editing someone's genome. Okay, so contradicting effects. One example here was just the argument of episodic memory versus working memory, and the decrease versus Alzheimer's. Again, you can look up on some of these if you'd like to. Deletion of CCR5 gene is a big one because one thing I can point out is that a lot of genes tend to be a catch-22. Like classically, I think. like the genes that are related to inflammatory bowel disease are also heavily linked to disease resistivity. So, you know, if you have a higher risk of colon cancer, you have a better disease resistivity. So it's a trade-off. You know, in this case, CCR5 confers resistance to HIV, but would also increase the risk of West Nile if we just simply delete the gene. So a lot of times genes have unintended consequences if we're not careful. But next is, again, Crohn's disease, type 1 diabetes, mellitus. This one is amazing. I'll try to talk about that in more detail. But again, just to check the time really quickly here to make sure we're staying on time. So I'm just going to very quickly go through the mouse model for PCSK9. This is believed to be a LDR receptor antagonist. In this case, cause of autosomal dominant hypercholesterolemia, excuse me. In this case, we're looking at a gain of function mutation for highly elevated LDL levels. So in this case, in this research experiment, they did a single loss of function mutation and it resulted in LDL levels 30 to 40% and an 88% risk in coronary heart disease or constructive heart disease, excuse me. Next is a two loss of function, a gain of function, pardon me, or two loss of function. Sorry, trying to go too quick here. Let me slow down here. Two loss of function mutation, which resulted in an 80% reduction in LDL-C. And the big thing is that there was reported no adverse clinical consequences, which again is the big concern of using this Cas9. So here we're just looking at the gel electrophoresis. Again, let me make sure I have my little cheat sheet here if I need it here. Okay, so we're just looking at a couple things here. A mock transfection uses control. They have a guide strand that recognizes exon 1 and a guide strand that recognizes exon 2. Okay, and the only one that's showing any type of cleavage bands in this case is the one that recognizes exon 1 and the exon 1 locus, which is why when we look at the exon 2 locus, they all look like the control because we are no longer recognizing exon 1. And as this paper describes, exon 2 was not provided with Cas9. So this one here, exon 1, has Cas9 in the guide strand. This one here, this package, has only the guide strand. So the one that recognizes exon 2 is not capable of catalytic activity, which is why all three of them looked like the control when exposed to the exon 2 locus. So the only one that was being recognized was the one that was capable of catalytic activity in exon 1. Again, they looked at it after 3 days of treatment. They had two controls, two GFP controls, and two exon 1s. Okay, so we're no longer looking at the guide strand for exon 2. Both of these recognize exon 1. And sure enough, they proved a cleavage band for both of these. Okay, pretty clean work. Okay. In this case, their measurement of the results was they were just simply looking for percent of mutagenesis. So they were just proving this result by saying, hey, we have cleavage in both of these lanes in comparison to the control, and they were proving that this technology does indeed work. Okay. Once again, they were doing another control 4 days after mouse liver. This was with no virus or no adenovirus being used. This is with the GFP adenovirus, but no Cas9 protein. So sure enough, we see that it looks identical to the control. And each of these lanes here are for the, has a guide strand and Cas9 for exon 1. So this is for exon 1, this is for exon 1, this is for exon 1, etc. And sure enough, we see a positive result in each of these lanes. Next, they just looked at off-target mutations. They compared the mouse that did not receive adenovirus to the mouse that did receive adenovirus and the exon 1 guide strand. And they noticed that off-target mutagenesis was indeed minimal, okay, or non-existent at all. So pretty awesome stuff. Okay, as they moved along, and again, you can look at the scores that they used. They showed that there was a reduction. I believe this first one here is, yeah, so this just shows you plasma PCSK9 levels. They show a reduction in PCSK9. They showed a control of triglycerides upon all three data points. And sure enough, a reduction in total cholesterol levels in the mice that had positive Cas9 activity. Okay, so this was, at the time, 2018, I found this to be a very impressive paper. Looking at this, they just also showed, what is this, ALT or alanine aminotransferase levels, showed that they're a little bit elevated, in this case in the liver. but otherwise considered non-toxic. Okay. Also, too, they use a GFP selectable marker in this case. They're able to recognize the GFP when looking at histological stains. Lastly, this is where the research paper is really interesting. So they did a Western blot, and they did no virus with respect to the exon 1 recognizing guide strand and Cas9. And they're looking at levels of LDL receptor, and they notice that the expression of the LDL receptor reemerges and re-expresses itself when the PSC-K levels were reduced. You have to remember again that if we go back and look, hang on, the PCSK9 levels were believed to be an LDLR antagonist. And they kind of proved that in this research experiment because the expression of the LDLR all of a sudden came back because it was no longer being antagonized. And here they're showing the beta-actin ladder as control, just to show you that the research or the results were consistent. Same thing here. They're just comparing it to GFP control and also a second set of exon 1 recognizing guide strands. Okay, so that's a pretty cool paper if you want to read up on that. I do have about 15 minutes left. I don't want to go over too many more models, so I'm going to kind of go pretty quickly through this next one here. This is for Duchenne muscular dystrophy. This is an X-linked disease, about 1 in 3,500 males. The gene to make dystrophin in this case. Large cytoskeletal protein, which is responsible for skeletal muscle function. And as you know, the diaphragm is skeletal muscle. So unfortunately, these males would have a mortality range about age 25. In this case, it just shows you the exons that they're targeting. There's a premature stop codon that's inserted. And sure enough, the researchers said, hey, look, there's a protospacer adjacent motif located right there in the 5' 3' direction. And the researchers said, well, you know what? We can target that gene. And that's what they did. When they targeted that gene, they were aiming for HDR, non-homologous end joining. I'm not going to go into too much detail here because I'd have to explain what these TESLs and T7s are being used for, but these are basically being used to prove positive HDR. The red represents positive HDR, blue represents negative result. So here we see that there was positive HDR in several of these lanes. And they tried to take a percent measure, about 41%, 2%, 17%, 16, 40. And they even had a lane that they suspected had 100% HDR. Now you notice there's also some that received that were either control or better. This is the control on the far right, pardon me. This is the undigested. But on the far left, there were some that received that they believe were due to non-homologous end joining. Okay, so in comparison to the previous study, this is a germline study. In this case, you see that they're inserting this Cas9 complex in the dichorionic phase prior to syngamy. And in this case, they're also providing it with a single-stranded donor. Now, why I like this paper is it proves something that I use when I teach my students today, which is we're comparing wild type of the quadriceps, soleus, and diaphragm and heart for the dystrophin arrangement. In the mutant, we see that it's almost completely absent. With estimated 17% HDR, we see the dystrophin protein is not only reemerging, it's actually starting to assemble itself in its primary conformation. With about 41% of HDR, we see that it almost looks functional, but here's where the paper was interesting. For those that didn't have a TESL band that was estimated to be non-homologous end joining, almost completely resembles wild type. So what this proves is that non-homologous end joining is very much okay, because the concern here was a premature stop codon. So we don't need it to be wild type. We just have to disrupt the stop codon. So non-homologous end joining was good enough, and this is what this paper proved. Here they take a laser dissection. They do gas chromatography. I'm not going to explain gas chromatography, but they look for a premature stop codon or a glutamine, glutamine, pardon me. Okay, now in this case, when they do the laser dissection, they were trying to prove mosaicism. And once again, this is a second paper that reconfirms that mosaicism is tolerated. In this case, they were looking for some that had GFP and some cells that did not have GFP as their selectable marker. And sure enough, this paper proved that mosaicism was tolerated. Interesting enough, they looked at the mouse studies here. We see this, they're showing grip strength with respect to serum creatine kinase levels. Of course, they're looking at males versus females with the standard deviation, so take some of this data with a grain of salt. But we see a definite improvement of serum creatine kinase levels in comparison to the percent HDR and also compared to wild type. I mean, we go from 6,366 to 350. So there's a significant reduction of serum creatine kinase, which implies that it's repairing that muscle tissue. When we look at the grip strength, we see the grip strength in this case improved as well over several trials. And you can make the argument that each of these results were actually enhanced. Again, you'd have to take the standard deviation into consideration and also the sex. But we see under all parameters that the edited genomes were actually better than wild type. So next is further concern. There's human embryonic studies, studying 3PN versus 2PN. So in this case, again, you guys can look this stuff up as well. This is just, I believe this paper had to do with the diagnostic processes. And again, you can combine it with the process we would use, even if we do the protocol at Austin Community College. We're still going to be using restriction endonuclease. This is going to be part of the primer design. But as you can imagine, you could probably use a collection of different protein complexes to achieve whatever your goal you're trying to achieve. Okay, and again, this paper is just trying to prove different levels of non-homologous end joining compared to HDR. How would you know that these are HDRs? Because these are ones that have the donated oligonucleotides. Okay, again, check time really quickly here. I'm almost done here. Just gonna wrap up a few more slides. Okay, and once again, they're just showing that off-target sites or off-target mutagenesis or indels are single nucleotide polymorphisms were minimal. Okay, so that's 3 out of 3. Really, in fact, there was actually 2 different sources that took here. So about 4 out of 4 papers presented showing off-target mutagenesis being minimal. So claims. Off-target reduced via using Cas9 protein, varying results across several studies. Mosaics reduced via co-injection Cas9 with sperm. Rumors fertility clinic in Mexico, right? Already doing mitochondrial edits. China claims, etc. So this is outdated, right? By now, this presentation is 7 years old. It's crazy to think that. So in this case, below are several contemporary CRISPR-Cas9 studies or clinical trials, diagnostic tools and detection of or inactivation of viruses, such as severe acute respiratory viruses. Anthropology studies such as reintroduction of archaic genes in the brain organoids demonstrated neurogenesis. That one kind of blew my mind. Knockout gene of embryonic stem cells to identify embryonic regulators. CRISPR or dead Cas9, which, as mentioned before, can be used as for epigenetic studies or histone remodeling. Cas13, which I think would be useful to the NASA team. Which is useful in editing targeting RNA strands. At NASA, if you did not know, they like to focus specifically on short reads for gene expression studies on the ISS and for ground control. In this case, it typically, you got to think, considering typically short reads sequencing targets random cleaves. Well, now we have the technology to start having site-specific mutagenesis to support that kind of research. Next is Cas7 through 11. These are responsible for RNA editing as well. They've been demonstrated at least, So again, take some of this with a grain of salt. But the big one's gene drivers. For those of you who know, there's this topic of gene drivers, and I recommend you learn about them because now we're starting to introduce reversal drivers, which really should be eye-opening if you take the time to go and look those up. Okay, look up Harris County, Texas. Okay, so personalized medicine. In this case, we've all heard that term, personalized medicine, just several different diseases here. You can kind of look over. I do want to pull out a couple ones I found interesting. This is through recent research, just kind of a couple days before giving the presentation. I want to kind of pull some contemporary studies. In 2023, the U.S. FDA approved first cell-based gene therapies for treating sickle cell anemia. This is a report by the FDA in 2024. These are the names of the products that you can look up, which are also can be used for editing hematopoietic stem cells. Also, cancer. There's several different T cell therapies, oncogenes, tumor suppressor genes, etc. You can imagine the giant use of this in the field of oncogenes. HIV/AIDS, about 9 out of 23 humanized mice treated with a combination of antiretrovirals and CRISPR-Cas9 had the virus become undetectable even well after a typical rebound period. In 2023, was proven safe and effective in 3 studies with no data presented. So once again, you got to take some of this with a grain of salt. But a lab in Amsterdam reported complete elimination of HIV from human cell cultures. And once again, this is simply a claim. The big one that most concerned about, I think this is great. Type 1 diabetes mellitus. There's one single patient that was selected for a phase 1 clinical trial in February 2022. So as you can imagine, this person is on her 3rd year. And from what I was reading originally, that the person was responding really, 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 really well to this therapy. So, I mean, the first thing I did is I called my dad. I said, "Hey, Dad, there might really be a cure for type 1 diabetes mellitus." But also diagnostic tools, those of you who are interested in agriculture or other studies as well, for those of you that might be studying or teaching microbiology, there's, of course, going to be several different uses. So other projects developed, there's, of course, the PASTE project, which this is amazing. CRISPR-directed integrases, which can correct large-scale aberrations by combining this with integrases and can head up to 36,000 genes. That's huge. Also, prime editing. This shuttles appropriate repair enzymes to the location of a nick rather than a break, and this enables a high degree of precision. So basically, imagine we're going to be using this to recruit other proteins such as, possibly methyltransferases or mismatch repair enzymes, etc. Okay. So limitations for gene therapy in 2025 is it requirement of a protospace or adjacent motif. A big one, the big challenge and concern is p53 induced apoptosis and cytotoxicity. That is not something we're not out of the weeds of, right? We still have to be concerned about that. In this case, toxicity for immunotoxicity or immunogenic toxicity or just cytotoxicity in general. Because Cas9 is not a small protein. In fact, one of the challenges provided is it doesn't fit inside of many viral vectors, which, hey, I have no problem with. I don't necessarily think it's a good idea to maybe put this in a respiratory virus. Okay, I don't think it's a good idea. Okay, so I'm okay with the fact that Cas9 doesn't fit in too many viral vectors. But moving on, efficacy and ethics. As you can imagine, we've talked about several previous concerns of parental autonomy has seen its first clinical trial in a disease of a protein lacking in a liver from an infant. So again, this just shows you some of the things I was looking at 2018. One specific is ex vivo. This is editing of immune cells and transferring them back into the body. This is how immune therapy would be done, and specifically knocking out the expression of the PD-1 receptor. In this case, we know that this is typically targeted by tumor cells to inactivate T cells. Well, imagine if we just deleted that gene and said, hey, our T cells no longer care. Okay, and that's the idea. Clinical trials, the early days, as I mentioned, beta thalassemias was a big one. Other gene therapies approved in 2017. Target rare forms of blindness and late stage, stage 4 cancer. So it's really impressive. As I wrap up conclusions, I just kind of want to show you the future. So this was a workshop I attended in 2023 with when I was employed at Houston Community College and with my alma mater at the University of Houston. This is known as CRISPR in the classroom. This can be done and completed by Donna Pattison. She's one of the deans at the University of Houston. And, if you want to, she will 100% travel to Austin Community College and do this workshop for our department. Basically, it just shows you the learning objectives here. If you'd like to read this, it involves primer design and also delivery of the Cas9 system and also doing gel electrophoresis for quality control. Very awesome workshop. I very much enjoyed it. I would recommend this for anybody to attend it. If you'd like to reach out to Donna herself and attend one of her future workshops, you can. But also, too, it was proven successful in the pilot workshop. I was a part of the pilot study. My own personal challenges, I would say, that would be difficult for community college students is when you're working with small quantities, nanograms per deciliter, it can be a little challenging. So just getting comfortable with working with small quantities, it would be something that the faculty would have to work with the students. Other opportunities, summer science programs. In 2025, I was recommended for a highly prestigious program that pairs educators with highly selected high school students. And they were teaching high throughput gene sequencing using artificial intelligence to quantitate data prior and following CRISPR-Cas9 protocol. Unfortunately, I was not selected, but I did not lose that opportunity to be motivated. As this last week, I participated in a NASA-sponsored high throughput sequencing training where I learned how to use artificial intelligence to spit out and do impressive work with real-time qPCR data, which is what is currently on the International Space Station and here at ground control. Long story short, we use aligners such as STAR and algorithms such as RSEN to spit out millions of reads in seconds. We can run FastQ and multi-C command prompts to analyze, instantaneously trim, and align your data. It truthfully is high throughput sequencing, and with AI, we no longer need to individually read millions of reads and analyze quality scores from raw data. AI will align this data to your parameters that you set with your databases and your codes that you provide it with, and provide you with also provided with an index, or database from databases around the world. And it will amplify as you sequence in real time. This applies to omics as a whole and can be used for transcriptomes, short reads, and genomics. This just shows you some of the data that I was looking at during the workshop. As you can see, we use a command prompt provided with the logic and the coding that we use to spit out this data. And I'm not joking you, it literally spit this out in 2 seconds. And it's reading millions of databases, millions of gene sequence and nucleotides, recognizing specific domains, all within seconds, and spitting this data back to you. For here, just showing you some examples of the coding that we were having to learn during the workshop and some of the type of very important data that provides. But the cool part is we can align this with the databases, both used by the ISS and at ground control, to use this data with either a fast QC summary or multi-QC summary, and this data truthfully is impressive. The goal, and as we continue to incorporate coding into the artificial intelligence and provide it with more logic and more information, we can get more impressive quality data sets. And the goal here is to have really alignable data that the astronauts can use. And this is going to be what they expect at NASA for you to provide really quality work. So if you can achieve this by both combining good technique in the lab, but also just a solid foundational understanding of artificial intelligence or just basic coding, you could really find a good opportunity in the field of biology. So the future is skills like Cas9 protocols are readily available, are very inexpensive, and is very easy to use, and primer design is palatable for the college level. High throughput sequencing and understanding of units like coding programs, R, Python is the future of biology. So arm yourselves with the tool of the future. And this was me when I attended the camp at the University of Houston.
And thank you very much, Daniel.
All right, yeah.
Really interesting stuff. I'm actually probably going to reach out to you and ask about maybe getting Donna's info, because that sounds like a really interesting workshop to have here.
Well, I appreciate it, Devin. Thank you for the opportunity, and I hope you all enjoyed. And I'm looking forward to the next presenter.
But before we let you escape, are you okay to entertain any questions if anyone has any?
Sure.
Any questions for Daniel? He just covered it all, huh?
Is the I have one question here.
Yes, sir.
Is the STAR like the STAR and the algorithm are available open source, or do you have to subscribe to them?
Yes, sir. Very good question. If you're interested, I can show you the camp that I was working with. If you get my email information, I will send you an invitation to the camp.
Okay.
Anybody's welcome to learn.
I will be, yeah.
Also, too. Yes, sir. Okay, so very important that you know this. In 2024, the ISS and ground control. So what it is, it's the research lab out of Ames, Iowa. It's the Ames Research Center. And in 2024, they actually made public all of the databases for the research experiments, using real-time qPCR, fast QC, and multi-QC data. So you can actually, especially if you have your PhD and you're looking to do a PhD project, you can actually request the raw data. But they have provided the multi-QC and fast QC databases for rodent studies, for several things that are currently being done, both here on Earth and for the ISS. So yeah, so this is very much open source. The idea is basically we've reached the point at NASA where they're now starting to resource scientists. The more people who can have looking at this data, the more people will be working with subsets, the better type of understanding of genomics and transcriptomics that we're going to be able to understand. So great question.
Okay. So I will contact you to figure out how to proceed with that.
Yeah. So there's star is not the only one. Arsen is not the only one. There's several different aligners that you can use. It's going to depend on the type of work you want to do.
Okay.
Mhm.
Thanks.
Yes, sir.
Any other questions for Daniel here? All right, y'all. Thanks for coming, and I'll see y'all at the next presentation. Daniel, if you want to stick around a little bit, I can make you the host. But if you're gonna take off, we can just cut it right here.
Sure, I'll stick around. How do I need to quit sharing for a second?
Let's see. I'll go ahead and stop the recording. I actually went ahead and just stopped it for you.